Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 19th January 2021. As we all know that we are revising the entire syllabus. Again, this is third round, and in this round I am solving one more test paper. Already we have solved one test paper in each chapter. This is test paper 2, we are solving it, and once we solve this we'll go for the case study cases, right? Already I have trained you in assertion and reasoning. We have answered the questions of assertion and reasoning type. In the next round we will take up that case study questions, wherein one paragraph will be there, questions will be there, you need to answer that. So here we go. Chapter number 2, sexual reproduction in flowering plants. So in this chapter, as usual, we are going to solve some questions which are of multiple choice questions, 1 mark questions, 2 mark questions, 3 mark questions, as well as long type 5 mark questions. So question number 1, a pea blast present in certain monocot embryo represents mesocotyle, B, second cotyledon, okay, rudimentary leaves, and scutellum. Of course this should be secondary cotyledon. 2, filiform apparatus present at micropylar part of the synergids help in A, providing nutrition, no, help in germination of seeds, no, help in absorption of water, and guiding the entry of pollen tube. This is the function of filiform apparatus. Question 3. The endosperm cells in angiosperms are endosperm cells, 3N. So haploid, tetraploid, and triploid, 3N, triploid, not diploid, of course. Question number 4. Parthenocarpy can be induced by the application of parthenocarpy, okay, fruit formation without ovary. So duration of light, temperature variation, of course cold treatment, recombinant DNA, growth hormones. Next, the fusion of male gamete with an egg in the embryo sac is called fusion of male gamete with egg. Double fertilization, of course, not the syngamy, autogamy, and triple fusion. Male gamete with egg apparatus, okay, that should be a triple fusion, egg apparatus. Now question 6, which are the 3 cells found in a pollen grain when it is shed at the 3-celled stage? Why is banana considered a good example of parthenocarpy? Why it is pseudofruit? 8, write the function of scutellum. Remember, recollect the diagram, vertical section of the seed. Then question number 9, in case of polyembryony, many embryos, if an embryo develops from the synergid and another from the nucellus, which is haploid and which is diploid? Next, what is self-incompatibility, opposite to compatibility? Why does self-pollination not lead to seed formation in self-incompatible species? Question 11. Why are pollen grains produced in enormous quantities in maize? Question 12. Define seed dormancy. Dormancy, sleepiness. Give the causes and significance of seed dormancy. 13. Draw neat label sketch of LS of an endosperm monocot seed. Again I said it's important that you practice lot of diagrams. Question 14. Differentiate between microsporogenesis and megasporogenesis. Which type of cell division occurs during these events? Name the structures formed at the end of these 2 events. Again diagram based question. Question number 15. Draw a diagram of male gametophyte of an angiosperm. Label any 4 parts. Y is sporopollenin, considered the most resistant organic material. Okay, so take a paper, start writing the answers, takes some time. Once you finish answering it, now compare your answers with the answers that I am giving. So first one, we said second cotyledon B. Yes, embryo of certain monocotyledonous seeds contain additional cotyledon besides one present in all monocots. This cotyledon represents the evolutionary stage of cotyledon formation from 1 to 2. Question 2. D. Guiding the entry of pollen tube. Filiform apparatus is finger-like projections attached with egg apparatus. This apparatus guides the pollen tube carrying male gametes for facilitating syngamy. Question 3. Triploid. Endosperm, of course, we said 3N, so this has to be triploid. Endosperm is the product of triple fusion in which 2 polar nuclei fuse with one of the male gamete. Endosperm provide nutrition to the developing embryo. Question 4. Growth hormones. Explanation. Development of seedless fruits without fertilization is called parthenocarpy. Parthenocarpy can be induced artificially by application of growth regulators. Okay, such as cold treatment, that should be wrong. So application of hormones is the correct one. Similarly, question number 5, it should be syngamy because triple fusion is different. That will result in the formation of endosperm. Here we are talking about the formation of zygote. So you need to have the clarity, you need to differentiate. Formation of endosperm is the triple fusion, formation of zygote is the syngamy. Normally we say syngamy fertilization. So this is fertilization 1 and that is fertilization 2. So we call it as double fertilization. Each pollen grain produces 2 male gametes. Okay, we say male gamete 1, male gamete 2, or male nucleus 1, male nucleus 2. One male gamete fuse with egg to form zygote. This is fertilization 1. The fusion is called syngamy. The other male gamete, male nucleus 2, fuses with the 2 polar nuclei. So 1 plus 2, 3 to form endosperm. That's why 3N. This fusion is called triple fusion and this should be fertilization 2. Since fertilization 1 and 2 are taking place, we call it as double fertilization. Right? Question number 6. One larger cell is vegetative cell and 2 small cells are male gametes. Question number 7. Parthenocarpic fruits develop without fertilization. Formation of fruit without fertilization. Hence they are seedless as banana. That's why banana is called pseudo fruit, false fruit. Question number 8. The single cotyledon of monocot seed is known as scutellum, single cotyledon. It absorbs the digested nutrition from the endosperm and provides nourishment to the embryo for its growth and development. Question number 9. Synergid embryo is haploid, means N number, and nucellular embryo is diploid. Question number 10. The inability of certain gametes, even from genetically similar plant species, to fuse with each other is called self-incompatibility. It is because of interaction between the chemicals of the pollen and those of stigma. So pollen pistil interaction. So if this is missing, then self-incompatibility arises. Question number 11. Because in maize, pollution takes place through the wind. Since it involves a lot of wastage, so pollen grains are produced in enormous number. Question number 12. The inability of seeds to germinate even if external conditions are favorable is called seed dormancy, inability to germinate. Seed dormancy may be due to one or more of the following reasons. hard and impermeable seed coat, sporopollen. Presence of certain inhibitory substances such as abscisic acid, ABA, coumarin, etc. Significance: it helps the seeds to tide over the temporary unfavorable period. It enables the seeds to disseminate safely and help them to germinate when the environment conditions are suitable or favorable. Of course, question number 13, very important. Many times this question is repeatedly asked in the board exam for 3 marks. Maize seed, vertical section. So you got the pericarp, endosperm, scutellum. This is what the question was, scutellum. And coleoptile, plumule. Of course, plumule is future shoot, radical, future root, and coleorhiza. So these are the parts that you need to label and understand the significance of each label. Write the significance in one sentence. Then you are thorough with the diagram. Practice and draw beautiful diagrams, and if possible apply colors also. Next, question number 14. Microsporogenesis, Megasporogenesis. Micro, it is the process of formation of microspores from MMC, microspore mother cell. Megasporo, it is the process of formation of megaspores from megaspore mother cell. Microsporogenesis, it occurs inside the pollen sacs of anthers, whereas mega, it occurs in the nucellus part of the ovule. In both the events, of course, what happens? Meiosis takes place. Microsporogenesis results in the formation of microspores, while megasporogenesis results in the formation of megaspores. Last question, again repeatedly asked. Sporopollenin can withstand high temperatures and strong acids and alkali. It is also not affected by enzymes. That much strong. Can, you just write withstand. Not stand, can withstand. Okay. Draw the diagram. So you got 2 things, germinative or generative cell and tube nucleus, which is having tube cell. Of course, vacuoles are there, exine, intine, and this exine will be having sporopollen. Okay, and germ 4. So draw the diagram, label it like this, and write the importance of each label. So we'll see next time one more chapter with test paper 2. Until then, draw diagrams, label it, keep practicing.